My name's Mrs. Tyler and welcome to Biology GCSE Draw Along Knowledge Organisers. Today we're going to try and condense a lot of information onto one sheet of paper so we can link our ideas together and then use it to read, cover, recite and do some good learning and test our knowledge at the end. Today we're focusing on some really important basic concepts. We're thinking about how substances move in and out of cells. So we're thinking about diffusion, osmosis and active transport. And we're going to spend a bit of time looking at the required practical too. So there's a lot there. If you need to pause the video, please do. If you're doing it at home, excellent work. If you're doing it at school, then please ask your teacher. They're probably marking, but they won't mind stopping, I'm sure. Okay, let's get started. So we have got quite a lot to fit on, like I said. So it's important that we set it out in a nice and ordered way. So today we're doing about diffusion, osmosis and active transport, but really we're talking about how stuff gets in and out of cells. So that's my title. Popped it in the middle. And we're going to, as always, divide our page down the middle. And then the left-hand side is going to be divided into thirds. My title wasn't quite in the middle there, but that'll do. And then this one, I'm going to extend across there. So I've got 3/3 there, 2/3 and 1/3 there. All right then. So in this section, we're going to be doing about diffusion. In this section, we're going to be doing about active transport. In this section, we're going to be doing about surface area to volume. And that's a ratio. Talk a bit more about that then. And then here we're going to be talking about osmosis. And this is probably the trickiest part of this topic. And it's got a required practical attached to it, which we know means that we're more likely to get asked questions on it. And lastly, in this part of the unit, we're just going to do a quick summary on exchange surfaces. Now, we are going to go into more detail about the lungs and the small intestines in other units. So we're not going to go into too much detail now. Exchange surfaces. Just a quick summary. Right then, let's make a start. So hopefully some of this will be really familiar and you're just like, yeah, shut up, carry on. And that's great. Some of it, though, might be a little bit trickier, a bit harder to remember. And again, if you get stuck, pause the video and zoom in if you can't see it very well. So we're going to start up here with diffusion. And we're going to start just by simply stating what diffusion is. And it's when particles move from a high concentration to a low concentration. Or lower concentration. So if we were drawing it like a hillside, diffusion would be, we've got lots of particles all together in a high concentration. And because they are moving, because all particles are moving, some will break through and they'll move around. This happens in liquids and gases, and they will spread out. And they will start to spread out. And that would be diffusion. All right. So they're doing it without energy. We say it is passive because the particles are moving anyway. If we wanted to think about that in terms of a cell, which we do because this is biology, Let's just draw one. So here we've got a cell. Cell membranes are permeable to some substances. That means some substances can go in. I'm going to show that as little holes in the membrane. So diffusion can happen over a membrane. In other words, into cells and out of cells. It also just happens in air and water, but it can happen over a membrane as well. So here's our cell, and around the outside of the cell, we've got a higher concentration of a substance. Let's pretend this is oxygen, for example. So we've got a high concentration of oxygen on the outside, lots and lots of oxygen, and a lower concentration on the inside. That means without that cell having to do anything at all, some of that oxygen will find its way across the membrane and into the inside. It will go down a concentration gradient. And it will keep moving. They're moving randomly, but eventually some will end up in there. And because they're moving randomly and some end up in there, it will do that until it reaches equilibrium, until there's the same sort of concentration on the inside as the outside. And it's still moving, it's just that the same amount will move out as move in at that point. So diffusion will happen until it reaches until it reaches equilibrium. So this is good. This is great for living things because this will just happen anyway. The cell doesn't need to do anything at all. And often it needs those substances. So obviously oxygen is needed for aerobic respiration. It wants oxygen to move in via diffusion. So that's great. But sometimes we need that to be really fast. We want to speed up the rate. So we can make it faster, otherwise known as increasing the rate, in the following ways. So the first way that living things increase the rate of diffusion is by increasing the surface area. We'll talk more about that in a minute, down here. Another way it can make diffusion happen faster is by making this concentration gradient, this difference between the higher concentration and the lower concentration, as big as possible. So increase the concentration gradient. The third way we can make diffusion happen quickly is by making the distance the particles have got to move as small as possible. We call it a short diffusion pathway. In other words, make it thin. And we can also increase the temperature, but living things really try not to do that because increasing the temperature denatures enzymes and damages the cell. So living things don't tend to do that. Cells and all living things tend to focus on these three things: surface area, concentration gradient, diffusion pathway. Let's have a little think then of the opposite of diffusion. So this is when particles are going from a high concentration to a lower concentration, and it's just happening because the particles are moving. It is passive. No energy is required. However, active transport, as the name suggests, is when particles move from a lower concentration to a higher concentration. And because we're going against the concentration gradient, this needs energy. It is an active process. That's what active means. It needs energy. So let's get back to our hill. We've still got high concentration up here. We're going to pretend this is inside the cell. We all want whatever's in low concentration down here, and it's like having to push it up the hill. It is active. It needs energy. Again, let's have a think about a cell, because this is biology and this is where we're thinking about it. Again, we're going to draw a cell membrane with holes in it, because cell membranes are permeable. Some things can just move through. So this time, we've got a higher concentration inside the cell. So let's pretend, I don't know, this is nitrates. This is a plant cell and this is nitrates from the soil. And there's not much nitrate on the outside, but we want it anyway. So the plant will use energy to get that nitrate inside, even though it's already got lots in there. Another example then of that is in root hair cells, the classic example. So root hair cells, remember, are this funny shaped cell. They've got square shape, and then they've got this long sticky out hair into the soil. And even though the cytoplasm is full of lovely minerals, it wants to take more in. So it uses energy, it uses ATP to take them in. So whereas this is down a concentration gradient, active transport is up or against a concentration gradient. And because it needs energy, we try to only do it when we need to. So that needs ATP. For those of you doing higher, ATP is the molecule needed to release energy. All right, so we've got our two main forms of transport in and out of cells. We've got diffusion going from a high concentration to a low one passively. We've got active transport going from low to high actively. And we need this process especially, but also this one to be as fast as possible. And like we said, one of the ways we can do that is by increasing the surface area. For some of you, this is going to be hard. Others, you're going to be saying, this is just maths, and that's fine. Let's just remind ourselves what we mean by surface area. By surface area, we mean the amount of skin or the amount of membrane, if it's a cell, that's facing outwards. Okay, so the surface area in biology is usually, if it's a cell, it's the amount of membrane, and if it's an organism, it's the amount of skin. How much skin or membrane there is. And it's measured, therefore, in centimetres cubed if it's an organism. If it's a cell, it might be measured in micrometres cubed. We're going to do it in centimetres cubed today. And the volume, hopefully you know what the volume means. It means how much cytoplasm in the terms of cells is inside it. And just like we might measure volume in litres, if we were having a drink, or a can of Coke, that's 330 millilitres, that's the volume. Cells are measured in millilitres or centimetres cubed or micrometres cubed. So it's the volume of the single cell or all the cells together if it's an organism. And that could be in centimetres cubed, if we'd measured the outside in centimetres cubed. If it's a single cell, it might be micrometres cubed. Okay. Here's the thing. Big things, despite being big, have a small surface area for their size. we say they have a small surface area to volume ratio. So let's have a look at an example. This isn't particularly big, but it's nice, easy maths. We're going to draw a cube. And that cube is 2 centimetres by 2 centimetres by 2 centimetres. If we want to work out the surface area, then we find out the area of one side and we times it by 6 because there are 6 sides in a cube. So 2 by 2 is 4 centimetres squared, and there are 1, 2, 3, 4, 5, 6 sides, so it's 4 times 6, which equals 24 centimetres squared for their surface area. The volume equals 2 times 2 times 2. Which equals 2 times 2 is 4 times 2 equals 8 centimetres cubed. Now, if we're going to write that as surface area to volume ratio, then we're going to write it as 24 to 8. If we want to simplify that, if we divide by the smallest number 8 on this side to make it equal to 1, 8 divided by 8 is 1, 24 divided by 8 is 3. So it would have a surface area of 3 centimetres cubed per centimetre cubed, 3 centimetres squared per centimetre cubed cytoplasm. Now that's all fine, but it doesn't give us much skin or much membrane to get oxygen in. Remember, cells and organisms need to get oxygen in, and the more membrane we have, the more space we've got to get that oxygen in. So the first thing we might want to do is increase the surface area. We could do that either by being small, so single-celled organisms often have a large surface area to volume ratio. Or we can make it so that some of our parts have got sticky out bits. Here's how this works then. If we cut this up, instead of having 1 8 centimetre cube, we had 8 separate 1 centimetre ones. Okay, I'm going to draw some of them into 3D. You don't need to bother doing that if you don't want to. Not really necessary. So I've cut this up like that. And instead of having 1 big 8 centimetre cubed, I've now got 8 1 centimetre cubed cubes. That means for these smaller things, or if I had 8 smaller things instead of 1 big one, the small ones have now got 6 centimetre cubed. Each side is worth 1 centimetre squared. So the small ones, each side is 1 centimetre squared, and there's 6 sides, which means it's 6 centimetres squared in total. The volume is just 1 because it's 1 by 1 by 1. Which means this smaller one now has a surface area to volume ratio of 6 to 1. So just by being small, it's still the same shape, but you can see it's doubled the surface area. So smaller things have got a larger surface area to volume ratio. So a mouse has a larger surface area to volume ratio than an elephant. And even if the mouse was the size of an elephant, all right, the small mouse would still have a larger surface area to volume ratio. But the more common thing that living things do is make sticky out bits. So instead of having 8 separate organisms, I might join those together, 1, 2, 3, 4, 5, into a block. 6, 7, 8. And suddenly, if I join those together like that, I've still only got the same volume here, because I've got 1, 2, 3, 4, 5, 6, 7, 8 squares, so it's still got the volume of 8. But now I've got the surface area all the way around the edge. I'm going to increase that surface area massively. So sticky out bits or long thin bits have a larger surface area than than smooth things. All right, tricky. On to the big one then, osmosis. And osmosis is just diffusion going from a high concentration to a low one, but it's just the diffusion of water. So it is only the movement of water. Just let me put this so it's straight. And unlike diffusion, it has to happen over a membrane. So diffusion can just happen in the air with perfume. When you smell something, that's diffusion. And it can happen in water. But osmosis has to have a membrane. And that membrane has to be partially permeable. It has to have holes in it to let some things through. So the movement of water across a partially permeable membrane and remember, we're talking about the water moving, so it's from high water concentration or higher to a lower water concentration. All right then, so we're going to start off by drawing our partially permeable membrane. I'm going to pretend we've magnified it. So here it is. This is our membrane. And it's got holes in it. Those holes will let small particles through, like water, but they won't let big particles through, like glucose or sometimes starch or proteins. So I'm going to draw some big molecules here. These are too big to fit through the gap. Okay, they can't go through there. They're in a solution though, which means they're dissolved in water. So I'm going to put some little dots around them. On this side, then, I'm going to put pure water, so it's just going to be all little dots. So this side has a higher water concentration than this side. There is more water molecules on this side than this side. Right. Which means the water molecules are going to go down their concentration gradient. They're going to move from this side, where there's lots of water, to this side, where there's less water. Now remember, this is a net movement. Some molecules will move forward, some will move back, but overall, more are going to move this way. Now, let's think of the consequences of that. It means if water's moving from this side to this side is going to have less water in it, which means it's going to weigh less. So afterwards, there will be less water, which means there will be less mass or less weight. And this side, equally, the one with the sugar in, or the one with the protein in, or the one with the salt in, water has moved into it. So there is now going to be more water, which means it's going to weigh more. And if we're thinking about the shape of the organism, as water moves out, if this is sealed in a membrane, this is going to shrivel up or shrink. And if that happens in a plant cell, we call it plasmolysis. Or PLAS.
That's in plants. And if it shrinks or shrivels in an animal cell, we call it crenation. So if this was a red blood cell, it might go from being a nice normal red blood cell to shriveled up and looking more like a raisin. On this side, however, it's got more water in it and more weight, which means it's going to swell up. That's called turgidity, or being turgid in plants. But for animals, this is bad news, because if water moves in and it swells up, the animal cells haven't got a cell membrane, and so they're going to burst, which is called lysis. So osmosis is really, really important, because water is moving in and out of cells all the time. And animal cells in particular, because they haven't got a cell wall, are at risk of crenation, shriveling up and dying, or filling up with water and bursting. Either way, it's not good. Now, the required practical was the one that looked at potato chips. So you would have got a series of 5 tubes. I'm just going to draw 2 here. And so you've got boiling tubes, you'd have filled them up with the fluid, and you'd have put a piece of potato in each. This piece of potato will swell up. I've drawn the sides slightly curved, and that's because it was put in pure water. So there's lots of water on the outside, which means the water moves in via osmosis. And because the water moves in, it's doing this side's going to swell up. So that was in pure water. Water moves in. Via osmosis. And it will swell up. We also put potato chips in different concentrations of sugar, and I'm going to draw one in a high concentration of sugar, and I'm doing it all wrinkly and shriveled because this one will shrink or shrivel. And that's because it's in a high sugar concentration. So this time I'm going to draw lots of sugar molecules around the outside and not much water. This time, water is moving out via osmosis. And so the potato chip is going to get lighter. Right, this time, we're pretending that this side is the potato chip, and as water moves out, there's less water, and so it will shrivel down. Plasmolysis will happen because it's a plant cell. Now, if we were going to plot the results of this is quite tricky because some of them get heavier and some of them get lighter. The change variable, the thing that we're measuring, which is percentage change in mass, in other words, how much heavier it got and how much lighter it got, will be positive for the heavier ones, but it will be negative here. So you have to draw it. So there's negative numbers there and positive numbers there. We always put the thing that we change along the x-axis, and the thing that we changed here was the amount of sugar, or the sugar concentration. And that's often measured in millimoles or moles per litre. This is 0 here. So let's pretend this is our scale. Notice it's going up evenly. So 1, 2, 3, 4 upwards, but when we're doing negative numbers, we're using the same even scale backwards. This is 0 here, which means there's no change in mass, but also there is no sugar at this end. And when there was no sugar, the potato chip should have swollen up. So we'd get the highest percentage change, it would get the heaviest. If we put a bit of sugar in, it will still get heavier. Some water will move in, but not as much. And if we put a bit more sugar in, it might be that still a tiny bit of water moves in, but not as much. But here we have tipped the balance. Now, at this sugar concentration, suddenly water is moving out of the potato chip, so it's getting lighter. So it will move down to this area. And this one is the strongest one. This one's like this one here, where it's all shriveled and it's down there. So I'm going to draw a line of best fit. All right. And that tells us that when it's in a low sugar concentration, this end, it gets heavier. But when it's in a high sugar concentration, it gets lighter, it loses weight. Right, that's quite tricky. It's also worth saying that at this point here, where there is no change in weight at all, no change in mass, that means that the concentrations are equal, which means the concentration of sugar on the outside is equal to the concentration of sugar on the inside in the cytoplasm, and so there's no net movement. And that tells you that that's the amount of sugar in potato cytoplasm. All right, so lots of hard stuff here. Right, and this is what you're most likely to get asked questions on. Osmosis, which is just water, not the sugar. The sugar can't fit through the gaps. And the water moving, changing the size and therefore the weight of cells. On to something a little bit easier then, just to finish off. And we're thinking about exchange surfaces. So how we can increase this rate of diffusion, how we can get more oxygen into cells or more carbon dioxide out. And this means they need to be designed to increase the rate of diffusion. Make diffusion as fast as possible. And so cells or organ systems that do diffusion always, always, always have a large surface area. Like we said up here, they have a concentration gradient which is maintained because we don't want it to reach equilibrium. We can't have the same amount on the inside as the outside. So it's constant or maintained. And lastly, they are thin. Short pathway. So quickly then, I bet you can think of at least two exchange surfaces, so parts of an organism that have a large surface area to let something diffuse in. First one then we're going to quickly recap is the lungs in mammals. And the lungs in mammals, instead of just having one huge alveolus or one huge lung like this, we have lots and lots of small ones because it increases the surface area. So instead of one big balloon, we have lots and lots and lots of little ones. They look more like a bunch of grapes. All right. So they all hang together and there'll be lumps hanging out this side. I've drawn it like it's cut in half, but there'll be lumps at the front and lumps at the back. Lots and lots of little alveoli. Each singular one then is called an alveolus. And they are wrapped in blood vessels. So we've got lots of blood vessels. So these are a bit rubbish, sorry about that. So these are the capillaries. Remember, capillaries are only one cell thick themselves. So because there's lots of single alveoli instead of one big one, they have a large surface area. And because the alveolus or alveoli have only got thin walls and the capillaries have only got thin walls, there's only a short distance. And because the blood is constantly taking any oxygen away that goes into the alveoli, so the blood is constantly taking it away, it never reaches equilibrium. It's not like the oxygen that's in the air in your alveolus ever reaches the same concentration as the oxygen in the blood. So the blood moving and the oxygen being breathed in and then carbon dioxide being breathed out at a higher concentration maintains that concentration gradient. So breathing and blood moving make sure it doesn't reach equilibrium or maintain the concentration gradient. Squeeze it in. All right. The next one then we're going to talk about is your intestines. And these have lots of villi. This time they're not microscopic. You can actually see them. It's a bit like looking at velvet. Okay. And again, they've got lots of sticky out bits. So we've got lots of surface area. It's a bit more like this time. And this time the blood vessels are on the inside, not around the outside, like they are on the alveoli. But there's still a lot of blood vessels going up into those villi. So here we go, lots of capillaries. So these are called villi. These are the capillaries. And again, we've got, just like here, lots of sticky out bits, so we've got an increased surface area. Thin wall of the villi, thin wall of the capillary. And again, we've got a good blood supply, and that blood is moving the food away. So this time the villi, sorry, I didn't say what the villi did. They're taking in digested food, so they're taking in sugar and amino acids. This is the inside of your intestine. They're going into the blood, and that blood is carrying it away, so it maintains the concentration gradient. I'm just going to put keeps concentration gradient. Last little bit then, which we need in less detail, is in fish. So fish have got gills rather than lungs, but there's similarities. They're a large surface area. There's lots of sticky out bits. So here's one sticky out bit. It's called a gill filament. And then there's sort of one sticks out beneath it. And then another one will stick out underneath that, and so on and so on and so on down. And they, just like the villi, are full of capillaries on the inside. So these give a large surface area. They've got lots of blood supply in them to take that oxygen out of the water. It's going to go into them, maintaining that concentration gradient. So again, we don't need to write it out, but we've got an increased surface area, we've got thin walls, and we've got a good blood supply to maintain the concentration gradient. And that is that. Look at that. That's a lot of stuff there. And so now your job, hopefully some of this you knew anyway, there might be one or two bits in there that are new. So your job is to go away, reread it, maybe cover, and try and recite it a couple of times. So diffusion is down a concentration gradient till it reaches equilibrium. Down a concentration gradient till it reaches equilibrium. Cover it up. Say it to yourself. Down a concentration gradient till it reaches equilibrium. And then we can make it faster by increased surface area, increased concentration gradient, short diffusion pathway. Increased surface area, increased concentration gradient, short diffusion pathway. Say it to yourself in your head. So you're going to go through each of those, read, cover, recite, and then I'd like you, please, to have a go at an exam question. See what you can remember. The exam question is going to be more application, probably particularly on this or this. But you know the stuff. You know that diffusion happens by itself. It's passive. Active transport needs energy, and that's why it's called active. I'm going to put a big zigzag around that. And organisms evolve to have different shapes to get more diffusion happening. All right, that's it for today. Well done, everyone. I'll see you next time.